Oh, there he is. Oh, what a voice. Now recording. All right. Today we are going to be talking about the precise phylogenetic analysis of microbial. Oops, that is the wrong paper. That is the paper that I followed to try to understand this paper. I'm coming back. I'm coming back. Um, oh, that just I spent the last 5 minutes reading the wrong paper. Awesome.
Time for a go around.
Okay, so yeah, that paper was about the software they developed to do this paper. Okay, domestication of local microbial consortia for efficient recovery of gold through top-down selection in airlift bioreactors. That's quite the sentence. Does anybody want to try to parse that before I break it down?
Using mystery goo to get metals out of garbage left behind.
Oh, okay. Well, that's the microbial, and you've got the metal, getting the metal. Um, So when it says top-down selection in airlift bioreactors, these are two important concepts. Every single part of this sentence is really important. So a top-down selection, it means we're instead of coming into an ecosystem and building up your that biological apparatus. I don't know. Your crew of plucky misfits from the ground up. You actually select from the native species. So not only is this paper about finding bacteria for gold mining, it's about utilizing native and indigenous bacteria, something that they're very, very keen on. And they explain why too. Um, So, the whole, hey, invasive species are bad for us and don't function as well has gotten all the way to the bacterial level when people are talking about mining. Okay, domestication of local microbial consortia. Tetra, you want to take a shot at that one?
Um, I mean, I guess
One
domestication of
one
local
word at a
microbial
time.
makes sense. I don't know, they're trying to harness the local bacteria for their own uses.
Yes, and they're farming them or herding them.
Okay, yeah, I can see how that would fall under that purview as well. Yeah.
And then there's one more word, and it's very important.
All right. Well, I don't know that word, so I think I may need some help on this
Consortia. Does anybody know what consortia means? Take a guess. Use your Latin and Greek.
Something about a bunch of someone.
Yes. Okay, so everyone knows what a consort is, right? It's like the person that's with you. Like if you go out to a fancy ball, your consort is your plus one. Consortia is the
Mm-hmm.
plural of consort. It's consorts, consortia, and it means basically all the homies going out together, not just going out on a single date. So it means so the full sentence when broken down means the farming and optimization of local microbial cross-species colonies for the effective recovery of gold using indigenous species in airlift bioreactors. And airlift bioreactors are basically just like giant sock slit extractors in my mind. I'm sure uh, somebody who knows more about mining than I do, here's a picture of them, might take offense at that or something. I don't know, but they're basically just sock slit extractors. They're just really big sock slit extractors. And for those of you who don't know what a sauce extractor is, it's a piece of laboratory glassware that's used for extracting oils and other materials from plant matter. So when you like want to make an extract of some sort of resin from a recreational plant and you want to do it science grade, in theory, like as an example. All right, we up to speed?
Yep.
Cool. 5 minutes later, we've made it through the title. Good job, team. All right. So a lot of this introduction here is actually just, or the abstract introduction is actually just a much, much longer version of breaking down uh, what that sentence means. Now, one of the reasons that they're using local microbial consortia is because um, the organisms that already live in the hostile environments have co-evolved with each other. And not only that, but we're calling these environments poly-extreme. So, when we talk about extremophiles, we talk about an organism that lives in a specific environment, which is basically inhospitable to other organisms. And usually it's something like heat or cold or acidity, something like that. So a poly-extreme environment means not just one, but multiple factors are outside of what we consider to be good for living. And the theory is that when you bring in other species, you basically have to, like, maybe you've got your favorite mining bacteria, right? Uh, and that's actually kind of a joke because everybody uses the same fucking bacteria. They're just kind of all co-evolved to their environment. But say you've got your favorite mining bacteria and you bring it into well, let's see. And this was done in Patagonia, which is awesome. So um so they ended up if you bring in your outside organism, you have like what's it called? I guess a refractory period, a lag. Well, your organism kind of gets the ever loving crap punched out of it by the ecosystem and has to evolve. Meanwhile, if you have already pre existing organisms that evolved, maybe they just need to be like wrangled into the proper situations to help you out with what you're doing, right? Like, don't just necessarily drag your favorite animal from continent to continent when there's, four-legged bipeds everywhere, right? Does that metaphor make sense? That analogy?
There were no horses when the colonizers arrived to the
There were
New
four-legged
World.
bipeds. There used to be a lot of four-legged bipeds in this area. I'm just saying it's our invasive species problem. We have a tendency to just find one animal that makes us happy and then just drag it all over the planet. Um and that has a destabilizing effect. Quelle surprise. Some things just don't work in other places. Um so like I said, everybody basically uses the same bacteria for industrial mining and mine cleanup because it's I will stop yawning at some point, I promise. Because it's easy and because everybody knows that it works, and it's called acidothiobacillus, which means it likes thiol rich environments that are also acidic, which, as you can imagine, is kind of exactly what you need. Um, so the interesting part about this is that um, you've got these acidothiobacillus, and they actually kind of live all over. But each region has its own like cultural flavor of bacillus, and so what they did is so like whenever you take things out and you try to culture them and you try to isolate them and stuff, especially when you're working on a paper like this, like you can't when things are working in concert, like in tandem in a consortia, um they play off of each other's strengths and buffer out each other's weaknesses, because survival is the name of the game and cooperation is the best way to survive long term for any species. So um so when you isolate these things, you don't actually see the entirety of how its genes are being expressed or what its abilities are, right? In the same way that if you have a monoculture crop, you get consistently the same food, but you don't get like what could be better food or the best food. You're going for consistency as opposed to optimization. Or you're going for optimization but in the wrong way, in the wrong area. Yield as opposed to quality. Um, so what they did is they did a metagenomic analysis. Um, does anyone here know about metagenomic analyses, how they're done? Anyone want to take a stab at that?
I mean, if I'm guessing blind, which I am, I'd say that they analyze the genomes of a bunch of similar things and look for larger patterns in the data.
That is one of the ways to do it. That's not how it's being used in this paper, but you have a piece of it, yes. Anyone else here know about metagenomics? Done any shotgun sequencing, stuff like that? Just on the off chance that random bio skill falls out. Okay. Um, so there's a very long introduction here that's just talking about like how important all these organisms are, what they're good at, how they're like um how they have like complex micro social ecosystems that um mean that they work together. Um a lot of them swap out like will have specialized genetic traits that only technically work with the other thing. So what they go out is what they do is they go out and they collect a bunch of samples. So if you go down to page 3, um, it really is like a giant list. They took samples from multiple mine sites, um, all in the same region though. Like you can see they've got the address there, the coordinates. Um, and then so they went through mines that were old, mines that were new, mines that had been handled poorly, mines that had been handled well. They really went everywhere, and they sampled everything. They took scrapings off of the soil, they took water, they scratched off biofilm, they took chunks of mine tailings, they sampled slurry. Like they really just grabbed like one of everything. um and then they figured out what kind of elements were in there, and and then they isolated out all the organics, um which is pretty easy to do actually, because what do organics have that what is like one exceptionally distinct thing that organics have that would make them easy to separate from inorganics? It sounds like a weird question, but it'll make sense in a second. Anyway,
Nitrogen?
uh, close, except for environmental nitrogen can be floating around as well. Nitrogen will bind to carbon, so like that's one of the reasons that people are like, oh, when you use biochar, expect a bad year. Like you throw the carbon in there, it binds to the nitrogen in the soil for slow release. The answer is lipids. Um, in your average soil sample, lipids are made by organics. Inorganics don't make lipids. So, and lipids are easy to separate because they do not like to mix with water. Um, so that's the simplest way of outlining it. Basically, you just separate out all the lipid associated material, and then you can just go for straight up inorganic molecules and atoms on the other side. Like step one, pull out all the you know copper or something like that. So they got it down to basically just the things that were alive, and then they made a bunch of bioreactors, and they grew them. They grew them all together in the same bottle. So like 20 years ago, 25 years ago, this would have been totally not something that you do. Um, Taking all of your samples and growing them together in one bottle instead of isolating them out into individual samples um, was just not done. We didn't have the ability to look at things without being exceptionally reductionist, which meant that if from a genetic point of view, since I think in many of our talks, it's been expressed that genes turn on and off given environmental conditions, given proximity to other things. So it stands to reason that our actual understanding of the functioning of these organisms is flawed by the fact that we are observing just the organism and not the complex system in which it works and has evolved inside of, right? Yes, that makes sense.
Yes.
Okay, thank you. All right. So then they just made like they had a word for it, and it wasn't like goo, but it was like goo. Anyways, they made their consortia, and oh yeah, they called it the Relave Viejo slurry because it's the slurry of bacteria that they made from the the Relave Viejo mines. Um, all right, so then they used a very simple kit. It's the DNA soil extraction kit. They're like $75. Everybody uses them, and then they pulled out all the DNA that they could, and then they just sequenced it all. They didn't, which is like a very different way of sequencing than the way you normally think about it. They weren't looking to necessarily put anything in order. They were just looking for chunks. Any chunk that was long enough to like be defined as a thing got put into its own little box, right? Every time they picked up a sample, they would put it into a box. So imagine reconstructing a chandelier. Every little piece goes into its own little box, and then you array all the boxes out. Technically, you have a full picture of the chandelier. You just don't know how it's arranged. But at least you can see all the pieces. Like if somebody was to ask you what the chandelier is made out of, you are going to have a better, more concrete answer after you've taken the chandelier down and looked at all of its parts. It doesn't actually need to be a functioning chandelier for you to explain how it works, right?
Yes.
Yes. And then so uh, uh, the next couple of pages are mostly just about um, experimental design, like how they made their slurries, how they got everything else, and then they also have um, uh, these native species. And when they started the native species, they actually started them in fairly mild conditions, and then they kind of punched them back into shape by hitting them with uh, the acidic conditions and the toxic conditions of the normal vine, and then they waited to see what evolved out. And uh, like they had mentioned, it you can kind of see the acidophilus or the acidothylbacillus. Ugh. Um, did I just like ruin that name again?
Yes.
Thank you. Somebody else wanted to say that so I don't butcher it. No,
Uh,
it is. It's acidothylbacillus.
oh,
Ha.
that's correct. Yes.
Great. And then they did all of the standard tests because, like, remember, this is about bioremediation or oxidizing metals, is what it really comes down to. And so they ran the all of these different tests to see how they worked, and they had their consortia, their like faux consortia, and then like their negative control consortia, and then consortias from two mines, the RVS and the BVS. And you can tell how much of the metal got solubilized by the microbial consortia after 60 days here down in table 1. And so um, for those of you who don't know the process of what's trying to go on right here, um, the in this situation, this is like a really kind of bullshit way of biomining, in my personal opinion. The gold mining process is exceptionally rough. It requires a tremendous amount of thiols and also really really low pH to turn the gold into solution. The simplest way of explaining how all this is done is basically they take the mountain, they gouge out the parts that they like, and then they try to aggressively turn all the metals into liquid so they can strain them off using lots of like sulfurs and mercuries and um, acid. And then the gold is then isolated from the remaining metal, and the rest of it is kind of dumped in a big pile in the back. That track for everyone?
So they pour a bunch of acid into a mountain and
Yeah.
they
Yep. So the uh, the acidothylbacillus has both its acidos and its thiols. It's in the name. And then what they do is they basically just spray these things on a side of a mountain. So instead of spraying sulfur or acid on the side of a mountain, they spray a bacteria that makes an acid on the side of a mountain. See, that's way greener. Because they're not doing it, the bacteria is doing it, and that means it's natural. Right? That's how that works. Um, so that's the actual process. Like if you were looking at it, you can see these pictures. It's basically like they strip the side of the mountain, and then they run drip lines like on the mountain, and then they just inoculate, and then they come back later. Of course, this method is talking about the airlift bioreactors, which is a little more step intensive, but basically comes down to the same thing. You take all the minerals, you put them into one of these bioreactors, which is probably, I don't know, they're very large. They're industrial vats, like the size of buildings, um, large buildings. And then they stir it around there with the bacteria, and they make sure it gets enough oxygen that the reaction, because it's an oxygen dependent reaction, and so they make sure it has enough oxygen to run the process, and the more bubbles you get, the faster it goes. And but it's still they still end up with the same waste process. It was just made by bacteria instead of people.
Is there
Not my
another
favorite.
bacteria that can reverse that?
Like another bacteria to go out there and base it up.
Yeah.
Yeah, or we could just neutralize it before we dump it out. Um, but 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 that's like, that costs money and time, and that would imply that, you know, well, why do you nobody wants to spend money on garbage. In theory, bioremediation is basically our attempt to do this thing that you're speaking of. You know, this giant pile of waste, and then you counter it with some other organisms. The thing is that you need to find an organism that is making the counter. So if you have something that's very, very acidic, you want something that's making something basic to balance it out, right? Except organisms that make basic environments do that because they live in that environment. It's not even a because. It's basic organisms live in basic environments because they make the environments basic. And the environments are basic because that's the environment they live in. It's kind of like a feedback loop. So it's very difficult to get a bunch of basic organisms and have them take over an acidic environment or even balance it out. People generally just try to neutralize or lock down those things, or most usually just ignore it because it's somebody else's problem.
Are there any good choices for how to make it more basic, like a calcium carbonate?
Uh, working with a team on a project for that right now, actually, for both removing materials from mining waste, which is where they and also rehabilitating mining waste so that it can become something like soil. Because right now it's seriously like super freaking hostile. And it's radioactive. Don't forget that there's always a little bit of radioactivity in those mine waste sites. All right, so that's enough about that, though. Does anybody need more clarity on this method? It is fairly simple so far.
I'm aware that they just took a load of things. They took the genome all over it. They took the genome bit, and that's pretty much it. Correct?
Correct. Yeah, all of that boils down to we took a bunch of stuff and we sequenced it all as best we
Yeah.
could. We sequenced parts of it. So then they used a really clever piece of software, um, which was the other paper that I was reading and got distracted about when I like read off the title of the paper and read the wrong one. Um, and they basically cross-checked all of their little pieces that they had sequenced against like every known genomic database, and started. We had all of these sequences in boxes. Remember, they isolated them out to boxes. Now they use this to start to cluster the boxes so that you can see like where these genes came from. While we do share a lot of genetic material with lots of things, that percentage that is different is very different, and you can use very easily targeted primers to start to hunt down these things. There's always at least one gene that makes an organism distinctly itself. And so what they found was while the so here we get this paragraph. It's called Temporal Variations in the Diversity During Domestication. So because they're consistently trying to push it towards a certain environment as opposed to an environment that's at least a little more in flux, like it would be if it's outside. They did see some shifting. There's the Acetothiobacillus uh, Ferrovum. No, that's not right. That's the wrong Pharaoh. Well, a lot of the Acetothiobacillus were kind of like the winners. They definitely outcompeted a lot of the other things. But you would see fluctuations because a lot of the metabolism that's being done by these organisms is due to the oxidation of iron and or the reduction of iron. So it's utilizing the iron as part of his metabolic system, and so you'll see these numbers start to cycle and make that sine wave that we associate with um, foraging pack dynamics, I guess is the best way to say that. And so when you get down here, figure 4, they're starting to collect their data, and you can see a little bit. They've got everything broken up by phylum, class, and order, which is actually pretty freaking fantastic. And you can see the percentages of each of those there. And so a lot of this is just talking about how they're seeing such a high amount of the acetylthiobacillus and how other things start to trail off. Trying to find the part that talks about. Anyway, the part that's super cool, the part that makes this paper kind of really exciting for me is figure 6. Everybody can join me there. I am trying to make more room on my screen. It's not going to happen. Okay, so this is. So you guys have seen like plasmid maps or genome maps before, right? Not necessarily meaning you've read them, but you've seen something like this circle before, right?
I think they were on
Much
biomass,
less
right?
dense, but yes.
Right.
The compiler.
Can you say that again, please?
I
Hello?
think they show up in Genome Compiler from Justin's old videos.
Yeah, in SnapGene. Yeah, so each one of these rings are all the genes that they've taken from from specific strains, and you can see the strains are lined up here 1 to 7, and then each ring has been labeled 1 to 7, and they have important sites of conserved usefulness, and they've been all lined up. So up here at the top, the one that says Defense GI, this is just like a standard protection mechanism, and you'll notice that it is conserved all the way down to type 5, and then type 6 and 7 don't seem to have a lot of that. In the heavy metal resistance, again, we're seeing a lot of so the first two strains are wild strains, and then 3 and 4 are the ATCC strains that you can buy online. They're based off of them, and then I'm pretty sure that 6 and 7 are non-native types, and 5 is another mine site. Anyway, so you can see this heavy metal resistance gene, the magnesium uptake gene, which is important for its metabolism, and also stress protection. So basically, if you want to be a powerhouse of acidothioBacillus, you have all of these genes. Sometimes more, even more versions of it, or bigger versions. You'll notice that GDI 221 here has a tremendously large magnesium uptake section, which is probably really helpful. They don't outline why things are bigger or smaller, other than there is more of it. And so a lot of these, especially these sections, they put forth the concept that these are horizontally transferred. So these are things that work and then have been swapped in between species, because it's so extremely functional that it just does. And I'm sure you guys have heard about how bacteria will scavenge the bodies of the dead for useful pieces of genetic information. Do you guys know about this?
Yeah, you've mentioned that before.
Yeah. What I really like about this, one, is showing how well everything fits together. But two, they actually put the genes, like the actual sequence of the gene, next to what they're pointing at, which means in theory you could actually just take, isolate these 4 genes and then smack them together, and or use them in other situations. We're actually going to be selecting for these 4 genes, and then doing some genetic modifications to see if we can increase our heavy metals resistance, especially in organisms that have different mechanisms. If we can get them to borrow some of these mechanisms, it'll be fantastic.
So does the light part of each circle going down into the middle represent how it's conserved between species?
Correct. And if you look around, you can see that there's a fair amount of that as well. And so we said species, and I'm not 100% sure if species is the best word there. I'm going to go, yeah, I guess maybe
Would be a better
family.
word. Oh,
I'm trying to
family.
think. So species is, I guess species is the right word.
They
I was going to say family, but
they're very sexual,
all
aren't
of
they?
these are the same bacteria. They're just different variations. So species is correct. No. All right, and their takeaway from the paper is that using indigenous species seems to work really well. It has an increase in rate of handling the environment, and there's a bunch of other random stuff that just seems to make it work that nobody really thought about because everybody was looking for just the most aggressive Acidothiobacillus. Um, part of the reasoning behind this is that the consortia kind of works as a team to protect each other from the conditions as they change over time. So, suddenly dumping 20 tons of highly acidic, mercury laden slurry onto a patch of ground is a very rapid and aggressive change. And so, working as a team, they're able to buffer each other. Each one of them likes the environment kind of in a specific way, and they help each other out. And this means that you have a slower die-off rate for your bacteria that you want to keep, which means that you get a better rate of oxidation, and this means you should be getting higher yields. This, of course, is what they have to make the takeaway on. But the most important thing that they're trying to talk about is how important the teamwork is for survival. So this is
Yeah.
this is this is a great intro to metagenomics paper because it doesn't go super heavy into the metagenomics. It's a black box. You are supposed to kind of understand how that works already. If people are interested, I can do a paper that's specifically about the metagenomic process if they want to continue learning about this topic or not.
When you say they work together, do you mean they can make a biofilm like I think Orison presented on earlier?
Oh yeah, I mean they talked about getting biofilms, but sometimes it can be something as simple as utilizing certain elements in the environment so that they don't become too much for another organism. Like I like there's lots of things that I like, but I'm not supposed to eat them all. You know, I can't just live off of jelly beans. And even if I was to be eating a healthy diet, if the ambient jelly bean content that has been slipped into my food is higher than normal, I will get sick, right?
Yeah, yeah.
Yeah. Another thing that they talked about in this was also that there's a bunch of other materials that are being isolated. So it's about gold, but you're also grabbing all of these secondary byproducts as well,
You mentioned
and either
radioactivity earlier.
trapping
Is
them
there
or
a
removing them. I'm sorry, what?
When you mentioned radioactivity earlier, is there an atomic gardening situation going on here
Um,
with bacteria?
Probably, but no more than what would be. It's natural-ish. I mean, atomic gardening was done with cobalt, and if you're going to get radiation in your tailings, cobalt is the first thing that I think of. So technically, yes, except the cobalt's kind of like pre-existing, it's just concentrated. And bacteria are they mutate so wildly at this level, it's more like a static variable than a confounding variable. Do you know what I mean?
It's always
It,
there.
it-
Failing to
it's
remove.
always there, exactly. So they don't discuss much about that. Actually they don't mention radioactivity at all. I just mentioned it because I know it's there, but it's not actually important to this process. It just happens to be a thing. But evidently it's not that serious because there's some guy in Australia who said he's going to mine me some gold mine tailings, and I haven't heard anything about paperwork yet, which is going to be interesting. We shall see.
Is that considered transferring gold from another country?
It is not. The gold is measured in parts per billion. I could, in theory, walk past a gold smelting plant and have roughly the same amount of particulate gold get in my skin and in my lungs. I'm not transferring gold at that point. That's my evil plan to destabilize an economy. I'm going to bring in 6,000 children on a giant field trip, and they're all going to. Everyone's going to take $1 when we leave. Ha! That'll show your economy.
Just as long as Nara and I can have their teeth. That's all that matters, Gabriel.
Well, that's what I
Of
was going to
course.
say. So you could have gold fillings, right?
Yeah,
Right.
there we
Yep.
go.
And and of course, that many witnesses, there's going to be loose lips, so it's best to just, break down your tools and put them away when you're done with them. Nothing? Wow. Okay, so any more questions about the paper? Is anyone interested in actually learning more about metagenomics more than me just like kind of casually mentioning that it's a thing?
How much of your lab project with this can you talk about?
My lab project? Oh, a bunch, especially because we're not to the genetic modification part yet, so I can't say what we're doing because we're not. So
Yeah,
what do you want to know?
that'd be
What do you
interesting
want to
to hear
know?
about. Well, I guess you had the salt and sea sample. Is that doing anything
Oh
yet?
gosh, yeah, all of those got destroyed. That actually created a second 10 Gentle project, which was about designing a standard field sample case. So that when some very enthusiastic newbies go out to collect samples, they don't just end up cramming everything into empty water bottles and then using vodka to sterilize their tools and then coming back and forgetting about everything and having it die.
It's very specific.
Yeah, well, it's like it happened. I remember it like it happened. Um,
Did you
yeah,
try any of the
so
USDA soil sample guidelines?
um no, the problem was literally just like the collection kit needs to be made closer to inebriated 20 something proof.
So
Like,
you think to have thousands of samples sent in to get a better representation?
oh, we're gonna be quantifying all of our samples. We've got uh LC and a GC, an atomic force microscope, and and
Liquid chromatography, right?
yeah, that's what an LC is. LC is
To
liquid, G is
expand out the
gas.
abbreviations for people.
Yeah, liquid chromatography and a gas chromatography setup. Um, we are going to be sending, like, you can get soil samples tested for like $7 here per sample because I'm in the Midwest, and so getting your soil sampled is like a a ritual that is done by everyone around here because farmers. So so yes, that. But in terms of like heavy metal concentrations, because we will be looking for stuff that's closer to the parts per million. Um, even even really low parts per million, where it's maybe like a decimal point off from being in the parts per billion, it's still acceptable. Then we gotta quantify it so we know how to throw it away when we're done with it, or neutralize it, or whatever. Separate rooms set up for storage. And then we are planning on isolating out all of our species just so we can see what we have, but then we will be keeping one big running slurry as basically the what if we just throw everything in a big vat control. Just kind of like my standard still yawning. My standard first test for every science experiment. You read a protocol and it's all like piecewise, and you're like, okay, but what if I just try to do everything at once? Almost never works, but sometimes you get really interesting results. Do you have any specific questions about the project?
What kind of products are you getting for those samples that are the species or bacteria or
Like
successful
what
produced?
what are we yielding?
Yeah, what form does it take? Is it like a sulfide or an oxide or like a lot of things?
Oh, no. One of the cool parts is that it's going to be sulfide free. Our units or our quantifier is atomic or elemental. Um, for instance, I'm very, very excited about the creation of gold nanoparticles using algae because it's so stupid simple, and the nanoparticle to gold cost breakdown is ridiculous. 1 gram of gold costs $80. 1 gram of gold nanoparticles cost $80,000, which is pretty nifty when you think about it. And so that just means taking gold in solution, exposing it to a certain process with organisms, and then having nanoparticles in solution, and they're elemental nanoparticles. Unless you want something different, then you use a different species. We've got a list of about 20 or 30 species right now. All of them uh, make a one or a series of shapes. So we've got pyramids, trapezoids, spheres, oblate egg shapes, and then the rest of it. But that's just some of the species. Those are the species that make nanoparticles. A lot of other species actually absorb the gold and then lock it down um, inside like a liposome. And then what you do is you use hydrothermal liquidification on your mass of algae, and you basically turn all of the organics into biocrude, and all of the metals get extracted that way.
How are you characterizing the product? Are you using the AFM to do that?
I'm using the AFM for two reasons. One, to characterize nanoparticles, but which is, like I said, only one of the things that can be done. But most importantly, the mechanism by which the gold is either caught and released into nanoparticles, because it is a passive process. The algae don't even need to be alive to make nanoparticles. They just bonk into them in solution and create them, alive or dead, which means that my theory is that there is some sort of surface formation that basically ratchets the gold into an elemental particle, Or
Or
again, like I said, depending on the species, you can make lots of other different types of nanoparticles. Silver, there's a gold ferrite, there's a nickel ferrite, there's a zinc, copper. Anyway, a bunch of stuff. Yeah, so I want to look at the surfaces of the algae and see what the configurations are, because if we start to do genetic modification or directed evolution that's going to increase the yield or rate, in theory, that would mean that it's increasing the number of residues on the surface that are causing this reaction. And since nobody really knows how the mechanism works, I just want to be able to look at it up close. And if we can find some sort of correlation between a mutation in the genome and an increase in residues, which gives an increase in rate, then that's some pretty good data.
is that why you were looking at the sequences conserved between the bacteria in this paper? Like those would be
Between the
the ones
bacteria and the what?
in the paper that you just presented. Are those sequences
Ye-
the ones that you want to amplify?
Yeah, yeah. And then we're trying to use algae instead of bacteria, though bacteria aren't off the table. They're just kind of like everybody's done bacteria stuff. But yeah, it basically the same thing. And we're going to be looking for those pathways that they saw in the bacteria and to see if they're conserved in the algae. And then, of course, we're going to take those pathways out of the bacteria and we're going to cram them in the algae and see if it makes them better or if it kills them. Good question. Anyone else have a question about this paper, or the project that, which is why I'm reading this paper? Okay, then I guess that's a no. I've been tagged here in voice and video chat though, a long time ago. Ever get an explanation for why fungus grows on sterilized mine tailings? Well, it's sterilized. It doesn't mean it's inhospitable. I
I think
don't understand the question.
early,
I don't understand the question.
an early paper that you presented on mine tailings research said that after they sterilized their samples, they had an overgrowth of fungus, so they chose to not sterilize their remaining samples because of that.
Oh, yeah. Um, I mean, that happens a lot when you sterilize stuff. Something does or doesn't get sterilized, or it gets contaminated from the air. And because you have a sterilized environment, things just run amok. It's actually really, really bad for certain biology experiments because things work weird in sterile environments. The you know the the physical structure of our gut doesn't even form properly if we don't have gut bacteria when we're like still in the womb.
Does
Okay.
sterilize include fungus?
Sterilized can include fungus, but fungus are sometimes tricky. I think the sterilization techniques that would have been most effective might have reacted with the materials, though. Like ETOH sterilization. I'm not 100% sure what that would be like on mine tailings. Probably not a good thing. Figure 6 is gorgeous. Are there thermophilic bacteria deep in the Earth's crust? Probably. I mean, it does get very comfortable down there. Cozy even. So I'm thinking about, oh yes, bacteria love to grow on recently sterilized instruments. That's why the laboratory standard for sterilization is ETOH. Like you basically just take whatever you're going to sterilize, you put it in a bag, it gets vacuum sealed, and then it gets like soaked in ethylene oxide for a certain amount of time. And then it comes out sterile as fuck and already sealed. We tried it with some hydrogels. It doesn't work with hydrogels. All right. Well, it seems awfully quiet. I guess chat is over. Thank you for coming out once again to another journal club. Again, please ping me if you want an actual in-depth breakdown of metagenomics. If you've been, if your interest has been piqued by, if nothing but the beautiful graphics, that's good enough for me. Yeah, so just like ping me or let me know in journal club, and I'll see you guys on Friday.